Good afternoon to everybody. This is the trial video on basic immunology. Video turn on OBS software. Now I want to start basic immunology. Basic immunology contains definition, immune system, definition of immunology, definition of immunity, definition of response. Role of the immune system. There is a defense against microbes, defense against the growth of the tumor cell, kills the growth of tumor cells, homeostasis, destruction of abnormal and dead cells. As for example, dead, red or white blood cells, antigen antibody complex. Immune system composed of three parts. First part is the organs, second part is the cells, third part is the molecules. Now I want to discuss on first part, that is organ. The organ of immune system contains tonsil, adenoids, thymus, lymph nodes, spleen, Peyer's patch, appendix, lymphatic vessels, bone marrow. Second part of the immune system contains lymphocytes. Lymphocytes divided into two types, lymphocytes and B lymphocytes, and natural killer cells also lymphocytes. Next, under cells, monocytes and macrophages. When the monocytes present within the blood vessels, they are known as monocytes, and when the monocytes comes into tissue area, they are known as macrophages. And granulocytes divided into three types, there is a neutrophil, eosinophil and basophils. Last, third part of the immune system, antibodies. It composed of immune system, that is molecules composed of antibodies, complement, cytokine, interleukin, interferon. There are two types of immunity. First one is the innate immunity, the non-adaptive. It is obtained in born. First line of immune response, relies on mechanisms that exist before infection. Second part of the immunity, second types of the immunity, that is the acquired immunity, the adaptive immunity, also known as adaptive immunity, that is obtained after birth. Second line, it controls the second line of response if innate fails. Relies on mechanism that adapt after infection, that handled by T and B lymphocytes. One cells determines the antigenic determinant. Innate immunity based on genetic makeup, relies on already formed component, rapid response within minutes of infection, not specific. Same molecules cells respond to range of pathogen, has no memory. The same response after repeated exposure, does not lead to clonal expansion. Innate immunity mechanism, mechanical barrier, that is the surface secretion, mechanical barrier or surface secretion. That is mainly the skin, acidic pH of the stomach, the part of the innate immunity. Next part of the innate immunity, lysozymes, basic proteins, complement, interferons. These are the parts of innate immunity. The humoral mechanism. Humoral means that is a soluble in blood. Is a soluble lysozyme, basic protein, complement, interferon are soluble in blood. Cellular defense mechanism, natural killer cells, neutrophil, macrophage, mast cells, basophils, eosinophils. Here pictures are given, natural killer cells. The nucleus is the larger in size. The cytoplasm is very small cytoplasm. And eosinophil, that is the nucleus is a lobe nucleus, eosinophil. Generally by that is a spectacular type. That is whatever spectacle we are. And the spectacular type nucleus is present. Neutrophil multilobe nucleus is present, and basophil that is the very rare in count in our body. This is the basophil, picture of the basophil, and the granules, basic granules are present. Eosinophil acidic granules are present. That is the pinkish granules are present. Neutrophil neither take up the acidic or basic. Acidic stain is the eosin, and basic is the methylene blue. And monocyte, here monocyte is present, that is the granulocyte. There is no granule in the cytoplasm. Here granule, basophil, neutrophil contains granule in the cytoplasm. So the granulocyte. For this reason, they are named after granulocyte. Adaptive immunity, second line response based upon resistance required during life. Relies on genetic events and cellular growth. Third one, response more slowly over few days. Is specific. Each cell responds to single epitope on an antigen. Has amnestic memory. Repeated exposure leads to faster, stronger response. Leads to clonal expansion. Clonal expansion means that is the numerous cells will be produced. That is the when memory cells is present, that is the immune cells will be produced more and forms colony. For this reason, it is named after clonal expansion, colony formation. What is the difference between what is the adaptive immunity? It is active and passive immunity. Active means when our body actively involved in antibody production, whereas passive, where the host, that is the recipient, does not produce antibody. That is the antibody produced outside the recipient. And suppose outside the recipient, if human beings receive the immune substance like the sera, like this, it contains the antibody. That antibody are produced in the horse body or monkey body or any other body, and then the antibody enter into the recipient. Whereas in case of the active immunization, the antigen is introduced into the body, and after that, in a response to antigen, the antibody will be produced by the recipient, actively produced. So here, the recipient body takes part actively for the production of antibody. Now, I want to discuss the active immunity, passive immunity. Natural is a clinical, subclinical infection. Passive immunity via breast milk, placenta. Placenta. This is the natural, not produced by human being. The clinical means suppose the disease, suppose the one patient or one subject suffering from the measles or pox, and due to disease, the antibody is developed inside the patient. So due to germ entry into the body, then against the germ, antibody will be produced. That is the natural process due to disease. And passive immunity, natural, that is the mother's antibody passes the passes to the suckling child via breast milk, or when the placenta is present, the immunoglobulin G enters to the placenta. Now, enters into the placenta. Mother's immunoglobulin G enters into the placenta because immunoglobulin G is smaller in size and it can pass through the placenta, whereas other antibody which is larger in size will not be allowed the through the placenta into the fetus. Artificial immunity, that is an active immunity, that is a vaccination that produced by a vaccination. And there are different types of vaccine, live vaccine, already you have known, oral polio vaccine, easy vaccine. These are the live vaccine, killed vaccine, different types of killed vaccine, bacterial vaccine, viral vaccines are present, killed vaccine, purified antigen vaccine, different types of vaccines are there. And the last one part is the artificial passive immunity. That is the sera, anti-sera, immune sera. That is, sera is produced in the horse body or monkey's body. Then blood is separated from their body, and serum contains the antibody. That antibody introduced and that is the ready-made antibody produced in the patient. Now I want to conclude up to this. Next, it will be discussed. Next part of the immunity will be discussed in the next part.